This session is about the cell organelles. Inside the cell, there are different parts which are performing their respective activities to keep the cell functionable. So these parts are called as cell organelles. In eukaryotic cells, the cell organelles are covered with membrane, which is not so in prokaryotic cells. Let us see which are the cell organelles. Number 1, endoplasmic reticulum. Number 2, Golgi apparatus. Number 3, lysosome, mitochondria, plastids, and vacuoles. So endoplasmic reticulum is a network of tubular structure between cytoplasm and nucleus for transport of material. Endoplasmic reticulum can be of two types: rough endoplasmic reticulum and smooth endoplasmic reticulum. When the granular-like structure ribosome present on the endoplasmic reticulum, it makes the surface rough, hence called as rough endoplasmic reticulum RER, and smooth endoplasmic reticulum do not contain ribosome. So ribosome are the site or a factory for protein synthesis. On the other side, smooth endoplasmic reticulum manufactures protein or lipid. So smooth endoplasmic reticulum takes up this lipid and protein to repair the plasma membrane. So this process is called as membrane biogenesis. Second major function of SER is to detoxify poisons and drugs in vertebrates. Our next cellular organelle is Golgi apparatus, described by Golgi, Camillo Golgi. So they are in the form of vesicles and cisterns. Their basic function is like movers and packers. So what they basically do is whatever materials are synthesized inside the cell or near endoplasmic reticulum, it helps in storage, modification and packaging of those materials into vesicle and transport it to the target site. The second major function of Golgi apparatus is it involves in the formation of lysosome. So that brings us to our next cellular organelle, which is lysosome. Lysosome is also called as the suicidal bag of the cell because it dispatches, it cleans the waste and foreign material inside the cell, including the worn out cell or repaired cells. And how does it do that? Inside lysosome, there are hydrolytic enzymes called as lysozyme, and these lysosome burst open, and these hydrolytic enzyme hence digest the foreign particles or the food material in case of amoeba and unicellular organisms. Next is mitochondria. Mitochondria are called as the powerhouse of the cells because they give power and energy for the cells to function in the form of ATP, which is adenosine triphosphate. And like nucleus, mitochondria also has its own DNA and protein. Next is plastid. Another difference between plant cell and animal cell would be the presence of plastids in plant cell. There are two types of plastids, chromoplast and leucoplast. So chromoplast are the colored plastids present in leaves, fruits, and flowers. Example, the leaf is green in color because of the presence of chlorophyll, which is helpful in photosynthesis. Whereas the other type of plastid called as leucoplast is found in the starch, oil, and protein granules stored in the fruits, leaves, or flowers. Like mitochondria, plastid also contains its own DNA and ribosome. Remember, ribosome is a factory for protein synthesis. Our last cellular organelle is vacuole. So vacuole is like a warehouse or a storage house which stores amino acids, sugars, and various organic acid present inside the plant cells to give rigidity and turgidity to the plant. In amoeba, the food vacuoles contain food item which is being digested and thrown outside the cell. Based upon vacuoles, we can classify animal cell and plant cell as in animal cell, the vacuoles are small in number or small in size but many in number, and on contrary, plant cell contains large vacuoles but they are one or two in number present in plant cells. So this slide will show you the structural difference between plant cell and animal cell. And here you will find the difference between plant cell and animal cell elaborately. So with this, we come to an end of our cellular organelles.